Good morning, everyone. My name is Charles Bai. I'm a lead application scientist from Vector Builder. Vector Builder is a leading leader in solution provider for gene delivery and gene therapy. And I'm very glad to give this webinar about the basics of in vitro transcribed mRNA, or IVT mRNA for short, from design to therapy. Through this webinar, you will have a really good understanding about how to design and produce potent IVT mRNA for both fundamental research as well as therapeutic development. So messenger RNA is the intermediate molecule in the central dogma. It is transcribed from the DNA and directs the translation of the protein. All of these three molecules have been developed into successful drug substance. And the most widely used probably are the ones derived from protein. And the most widely used recombinant protein is probably insulin. And other recombinant protein used are targeting are either ion channel ligands or, for example, antibodies. Using recombinant protein as a drug, normally they will have a really instant response. And when they were produced properly, they should have a really low immunogenicity for the patients. However, the challenge or the difficulty with using a protein as a drug is that most of them have really limited cell membrane penetration and a limited duration in the patient body. And the production for recombinant protein can also be complicated. On the other end of the central dogma, DNA has also been made into drugs. The best example would be gene delivery using viral vectors, for example, adeno-associated virus, or AAV. Using such viral vectors, you can achieve long-term efficacy or even one-shot for life kind of treatment. And these viral vectors can potentially achieve gene correction. However, the limitation of DNA as a drug is their slow response because they need to get to the cell, transduce the cell, and go into a cell nucleus, then being transcribed into mRNA, and then directs the translation of the protein. And at the same time, when the DNA is delivered into the nucleus, there is a potential risk of genome disruption, which will lead to activation or knockdown of certain vital genes. On the other hand, using mRNA as a drug substance, it has certain merits that neither DNA or protein has. First of all, as we all been very aware of, mRNA has been used as very successful vaccination molecule in fighting against the COVID-19 pandemic. And currently, there are numerous RNA-based clinical studies that using RNA for immunotherapy for cancers or protein replacement for patients came with a certain type of mutation. And when RNA is properly encapsulated in lipid nanoparticle, it really has good cell membrane penetration that protein does not have. And also, when mRNA is transfected into cell cytosol, the expression is very instant. Usually, you will observe the expression within a couple hours after transfection, which we will see a couple of results in later slides. And duration of the mRNA is also limited in human patient. That is because host cell will consistently degrade and recycle RNA in the cytosol. So that really increases the safety of mRNA used as a drug substance. And at present, there's no genome disruption or incorporation report for using mRNA. The difficulty or challenge of using mRNA as a drug is their low stability both in tube and also in cells. And also, of course, the difficulty to design potent mRNA molecules. So this is a simplified diagram showing the vital components of in vitro transcribed mRNA, which really try to mimic the vital components of endogenous mRNA. On the very 5' end, you will find a 5' cap, which is important for translation initiation and also non-self recognition. Without a cap 1 structure in mammalian cells, the foreign RNA will immediately be recognized as a foreign molecule and trigger innate immunity and immediately be cleaned out by the host cell. And on the mRNA molecule, there are two untranslated regions, 5' and 3' of the coding sequence. Although they are not coding any protein, but they are very vital in regulating the expression and stability of the whole mRNA molecule. On the very 3' end, there is a poly tail region. In the endogenous mRNA, the poly tail region is usually around 250 nucleotides, but it can be longer or shorter. And for endogenous mRNA, the poly tail region is very important for the mRNA to be properly exported from nucleus into the cytosol. And also it's important for the stability and translation efficiency of the mRNA. In the very center, you will find the coding sequence for the gene of interest. So to produce an in vitro transcribed mRNA, the very first step is to clone the in vitro transcribed mRNA vector or plasmid. On this plasmid, you will find all the mRNA sequence from 5' UTR to the end of the poly tails. And all these sequence should be following a T7 promoter. And at the end of this sequence, there is a unique restriction enzyme site, which is used to linearize this double-strand DNA circular plasmid into a linear form of the template. And this in vitro transcription template will then be mixed with T7 polymerase and the ribonucleotides A's, U's, C's, and G's. This T7 polymerase will recognize and bind to the T7 promoter and initiate the transcription of the whole mRNA sequence and generate this mRNA molecule. And because this is a linear DNA, the T7 polymerase will automatically fall off at the end of this poly tail, and which also terminates the in vitro transcription reaction. So on the design of this in vitro transcription mRNA vector, each component should be carefully designed. Let's start with the choice of the UTRs. Choosing robust and validated 5' and 3' UTR can really help enhance the translation of the mRNA. Let me give you a good example here. So in this case, we use a reporter luciferase called Gaussian luciferase or GLOC. And this luciferase coding mRNA sequence is flanked by 6 different types of 5' and 3' UTRs. And they are named number 1 to number 6. And all these mRNA were prepared under the same condition to the same concentration and transfected to 293 T cells. At 4 different time points post-transfection, the activity of the GLOC was quantified to reflect the translation of this GLOC mRNA. And when to compare the different function of the UTR, we can look at the comparison between the group 1 versus group 6. And you can see that the expression of GLOC activity is almost 10 times for group 1 UTR compared to the group 6 UTR. So that really demonstrates the importance of using potent, robust UTR to maximize the expression of the IVT mRNA. So next component is your coding sequence for the gene of interest. And proper coding sequence optimization can really enhance the expression of mRNA molecule. Here's a good example. In this experiment, we use a reporter mRNA that codes a firefly luciferase or FLOC. And first, we cloned the vector that will using the FLOC wild-type sequence and prepared IVT mRNA and transfected that IVT mRNA from wild-type sequence to the cell, 293 T cells, and quantified the activity of firefly luciferase at 4 different time points. And as you can see, the endogenous wild-type coding sequence for FLOC, its activity is almost negligible to the scale of this plot. And secondly, we used another optimized version of FLOC, which is commonly referred as FLOC2. And when we prepared mRNA FLOC2 coding sequence and transfected it into 293 T cells, the expression is much better improved from the wild type. It is almost 10 to 100 times improvement at the peak expression, which is 24 hours post-transfection. At Vector Builder, we developed a proprietary codon optimization method and apply that to the FLOC2 coding sequence and generated what we name as high express FLOC. And when we use this mRNA and transfect it into 293 T cell under the same condition, you can see that the peak activity is further improved from FLOC2 by another around 10-fold. And such comparison is also consistent in vivo expression. We injected 3 types of IVT mRNA coding firefly luciferase into mice, and you can see that at every time point post-injection, the high-express IFLOC2 always achieve the best expression in vivo, which is consistent with in vitro data. So last but not least components on the IVT mRNA vector is the polytail region. Long and stable polytail region can really enhance the mRNA stability and translation efficiency. And a simple explanation for that is that the host cell has a mechanism to clean up RNA in the cytosol, and one of the mechanisms is using an exonuclease that will chew up RNA from its 3' end. So put it simply, you can picture that when you have a long polytail, that will really slow down the chew up from that exonuclease, and which in turn increase the half-life of the mRNA. Of course, that is a very simplified version of that about how to interpret the effect of a long polytail. So in terms of the function of the long polytail in vivo, in cells or in vivo, let me show you a couple of really good comparison. First, in 293 T cells, we transfected EGFP coding mRNA with different lengths of polytail from 35 nucleotide to 70 and all the way to 110 nucleotide in their polytails. And at 2 time points post-transfection, you can see consistently that longer polytail had better expression of the EGFP. And of course, the one with 110 nucleotide had the best expression. And such expression phenomenon is also consistent in another eukaryotic system, that is zebrafish embryo, where you find the 110 nucleotide polytail achieved best expression in embryos. So if we take one step back, knowing that long polytail can support better RNA expression, but how difficult it would be to clone such a long polytail to a plasmid. If you have a good cloning experience, you will immediately recognize that if you are cloning a long homogeneous pure A region to a plasmid, that will make the plasmid rather unstable. And that is true with a conventional IVT mRNA backbone. In this case, we used a conventional IVT mRNA backbone and cloned a polytail region of different lengths to that backbone, and also planted the poly tail region with 2 unique restriction enzyme sites. And after cloning, we use restriction enzyme to free this polytail region from the plasmid and run that into a gel to see how intact the polytail region is on the plasmid. And here's the result. As you can see, when the polytail is around 60 or 80 nucleotide, you can still have a one unique sharp band, which shows that the polytail region on the plasmid is still intact when the polytail is below 80 nucleotide. However, when you extended that to 100 nucleotide or beyond, you start to see a secondary band or multiple or even a smear of smaller bands. That shows that a good amount of the plasmid containing such long polytail has already lost the intact polytail as designed. And when you use such plasmid as a template for IVT reaction, all the generated IVT mRNA will have a various length of a short polytails, which of course jeopardize the translation efficiency of those mRNA. So at Vector Builder, we developed a proprietary IVT mRNA backbone that is really unique in hosting long polytail. As you can see here, when the polytail is extended to 120 nucleotide, you still have a one sharp band on the gel. Even when the length is beyond all the way to 130 or 150, you don't see a very obvious secondary band after the major band, suggesting the plasmid is still stable, holding such long polytail. One step further, we use a plasmid that containing 120 nucleotide polytail to prepare an IVT mRNA. And from that IVT mRNA, we chop off its polytail and run that polytail into a gel. And you can see only one sharp band that is located at approximate position of 120 nucleotide, which suggests that on this plasmid, this 120 nucleotide polytail is still intact. One step further, we developed an LC-MS-based method to accurately determine the length of this polytail. And by interpreting the raw data, we found that the polytail on this molecule ranges from 119 to 129 nucleotide. And the median number of this polytail is around 123 nucleotides. So to make a really quick recap here, we talk about the vital components and their functions in IVT mRNA vector. And the vector builder provides comprehensive technical support for IVT mRNA design and cloning. Specifically, we can help you to choose robust and validated UTRs. We can help you apply the codon optimization and also use our proprietary backbone to host a stable polytail up to 150 nucleotide. So next, I would like to switch gear and talk about the production and encapsulation of IVT mRNA. On IVT mRNA, you can find most of its components derived from the template that is ranging from 5' UTR all the way to 3' polytail. However, the only components that is not derived from the template is 5' cap. So how is it added to the in vitro transcribed mRNA? There are two approaches. The first one is enzymatic capping. After IVT mRNA reaction, you will have IVT mRNA molecule, and that is mixed with vaccinia virus capping system and other substance. And this enzyme from vaccinia virus will add a cap 0 to the mRNA. And then it is further subjected to another enzyme called mRNA cap 2'O methyltransferase, and also the substance. And this second step of enzymatic reaction equipped mRNA with cap 1 structure. Specifically about cap 1 structure, it has two features. First, the first nucleotide of this RNA is methylated with the methyl group here. And the first nucleotide is also linked to a reversed methylated guanine by a head-to-head fashion. So the second approach of capping is called co-transcriptional capping. In this in vitro transcription reaction, the template is mixed with T7 polymerase ribonucleotides as well as a cap analog. And here, the dashed circle highlights the structure of this cap analog, which contains two nucleotides. And on the first base of the nucleotide is methylated. And also, the first base pair of nucleotide is linked to a methylated guanine, by a head-to-head fashion. And this structure is identical with the cap 1 structure found on the end product of enzymatic capping. And in this reaction, this cap analog will be used as the first two nucleotides in the in vitro transcription reaction, and all the rest of the RNA will be extended from here. So as you can picture, after the reaction completes, all the generated mRNA will automatically be equipped with cap 1 structure. Comparing these two approaches, you can clearly see that the co-transcriptional capping is more straightforward. It has only one pod reaction, whereas the enzymatic capping has 2 or even 3 pods of reaction. And by our testing, the co-transcriptional capping is even more efficient. However, co-transcriptional capping is also more costly. But at Vector Builder, we provide both capping approaches, and we can achieve good capping efficiency with both of the approaches. For example, in this case, the enzymatic capping yield 96.7% capping, and in this case, co-transcriptional capping yield 99.5% capping. And in our understanding, we don't think there's a one perfect capping strategy for all cases. And we would like to evaluate your case, your applications of the mRNA, and your timeline to suggest the proper capping solution for that case. And if you may recall, the 2023 Nobel Prize in Physiology and Medicine, which awarded the discovery of a modified nucleotide in mRNA, and which became the pillowstone for the development of a successful COVID-19 vaccine. The incorporation of a modified nucleotide in mRNA is very important. And here are a couple commonly used modified nucleotide. The first one is M5-methylcytosine, or M5C in short. And uracil can also be modified by flipping this structure surrounding this red line 180 degrees, that will generate a pseudo-uracil, which can further modified by methylation and generate M1 pasine. And in this experiment, we generated 4 types of different mRNA that were made with unmodified or modified nucleotide. And by comparing the modified, unmodified mRNA expression in 293 T cells with the one made with M1 pasine, you can see that the one with modified nucleotide had 10 times higher expression in 293 T cells. And such phenomena is also consistent for in vivo expression. In this case, in this experiment, we injected mice with 2 groups of luciferase coding mRNA. In the first group, the mice received naked mRNA, and you observe almost no expression after injection. And in the second group, the mice received a different quantity of encapsulated mRNA. And now you can see the expression of luciferase in vivo after injection. At 48 hours post-injection, we collected serum from these mice and quantified the 2 pro-inflammatory cytokine expression, IL-6 and TNF-alpha. And you can clearly see that the mice received lipid nanoparticle encapsulated mRNA at a much higher cytokine expression compared to the mice received only naked mRNA. In the third group, we used mRNA made with modified nucleotide and also encapsulated. Now you will find that the expression of the luciferase is even further improved. And also at 48 hours post-injection, the pro-inflammatory cytokine expression is also reduced when the mRNA is modified. So that really summarizes the 2 reasons of using modified nucleotide in mRNA production. First, it increased the expression of the mRNA, and second, it reduced immunogenicity of the mRNA. And we have already come across a couple results using lipid nanoparticle encapsulated mRNA. And to generate a lipid nanoparticle, you need to mix several different types of lipid with cholesterol and mRNA. And the figure in the middle shows the image of the lipid nanoparticle mRNA that we generated. And these nanoparticles are usually subjected to QCs that measures the diameter and also surface charge. At Vector Builder, we have developed a very comprehensive panel for QC testing for every step of mRNA production. Because of time limitation, I will not dive into details, but feel free to reach out to us if you'd like to learn more about mRNA QCs. And last but not least section of my presentation is to go through a couple really cool and also exciting ongoing research or application of mRNA. In the first example, this is a classic example of using IVT mRNA for a vaccination purpose. We developed 2 mRNA coding antigen A or B. These 2 are viral antigens. And we injected the mRNA in mice, and after 14 days of incubation, we collect all the splenocytes from these mice. And those splenocytes were further stimulated with protein antigen A or B, and to detect how many of the immune cells can really respond to the presence of the antigen. And as the result shows here, most the mice received the mRNA vaccine had a really robust immune response when they are exposed to the recombinant antigen, whereas the mice received the PBS had no reaction after the stimulation. And the B panel only shows the quantitative interpretation of this result. And we also try to deliver tissue-specific mRNA delivery. And to do that, we generated a luciferase mRNA and also encapsulated in lipid nanoparticle. And we conjugated a control antibody on this lipid nanoparticle that targets no antigen in mice. And after injecting this lipid nanoparticle in mice, you can see that there is a good amount of expression in liver and spleen, which means that most of the lipid nanoparticle are trapped and also transduced in liver or spleen cells. However, on our experimental lipid nanoparticle, we changed the antibody to another one that targets the antigen on lung cells. And after injecting this experimental lipid nanoparticle in vivo, you will immediately notice that the expression in liver is reduced compared to the control antibody, control dependent particle. And at the same time, you can detect the expression of mRNA in lung, which really demonstrates that by adding a conjugated antibody, the tropism of a lipid nanoparticle can be redirected from liver to lung. And in optimization of the lipid nanoparticle formula, we have been optimizing the use of the lipid in the formula of the LNP. Conventionally, you will use a PEG lipid to form the LNP, and this is a conventional lipid nanoparticle and achieve good expression in liver and in spleen. However, at the same time, it also trigger quite amount of TNF-alpha and the IL-6 pro-inflammatory cytokine expression, and that is because PEG lipid can be immunogenic in vivo. So to address that issue, we changed the PEG lipid to a PCR lipid in the LNP. And now, after injection, you can see that the cytokine expression is much reduced. Whereas the expression of the lipid mRNA in vivo is also reduced. So now we are working on now we are optimizing the formula of lipid nanoparticle by increasing the transduction efficiency of a lipid nanoparticle while maintaining low immunogenicity. In addition to in vitro transcribed mRNA, there is also research about using in vitro transcribed self-amplifying RNA or siRNA. siRNA can be amplified for limited cycles when it goes into a cytosol of the cells, and that is just a feature that mRNA doesn't have. And because that feature, you can inject much less copy number of siRNA to achieve the similar expression that need much higher quantity of mRNA. In this case, EGFP were made into both mRNA and siRNA format, and the siRNA was only transfected to a cell at the half quantity of mRNA. But at every time point, you can see that siRNA expression is much better than EGFP mRNA expression, and all the way till 21 days post-transfection, you can still see some siRNA EGFP expression. And that is also observed in vivo. So here is a result that we just talked about injecting mice with mRNA coding firefly luciferase. And if we change that firefly luciferase mRNA to siRNA, and now you can see that at 48 hours post-injection, there is still a good amount of firefly luciferase expression in vivo, which is absent when the firefly luciferase was delivered as mRNA. And when it is delivered as siRNA, the peak expression was kind of achieved at 6 days post-injection. And all the way till 8 days post-injection, you can still see the expression from the firefly luciferase. And if you may notice, the injected amount of this siRNA luciferase is only 1/3 of the amount of mRNA injected. So such feature is really important if you are going to take your RNA to the clinical stage, because the better, more robust expression of your RNA molecule in vivo means that you need a smaller amount of the RNA per patient. And smaller amount means that you can really reduce the cost in preparing the clinical grade RNA and also shorten the turnaround time of the preparation. So another form of IVT RNA is called circular RNA or circRNA. That is different from both mRNA and siRNA because circRNA is existing in a single string circular format, whereas both mRNA and siRNA are existing in single string linear format. And because the circRNA has no exposed 3' or 5' of RNA, it should be much more immune to the RNA cycling mechanism existing in a host cell. That's why you can see that although both circRNA and mRNA has kind of similar peak expression, but circRNA can really be expressed even on the 11th day post-transfection, which is not same as mRNA. So that was the last page for my presentation. I would really like to appreciate your attention to my presentation. And Vector Builder provides high quality and custom IVT mRNA production, encapsulation, and QC solutions. I'd like to receive any questions about my presentation, and thank you again for your attention.